I'm Maria Rescigno, professor at Humanas University in Milan. I work on cancer therapy. So it's like a Darwinian selection. When a cell is so fit that it's not recognized anymore by the immune system, this cell, it starts growing, proliferating, and becomes a tumor. When the tumor cell is escaping the immune system, so the immune system recognition, you can try to re-educate the immune system to recognize again the tumor cell as a tumor cell, and so to kill it. And this is what is called immunotherapy, and this is what I'm working on. We should be, in this way, allowing those patients that are not responding to immunotherapy to become responders to immunotherapy. We arrived now to a point in which we have something that might be working, and we want to keep this to the bed of the patient. We are very excited, but we need to go ahead. A cancer cell at some point escapes from control from the rest of the body. So a cell, it acquires mutations, and these mutations allow the cell to become a quick and a faster growing cell. This cell is seen by our immune system as a non-self cell, so as a cell that is different from the others. And the immune system starts to recognizing these cells and kills these cells and eliminates it. At some point, when a cell, a cancer cell, is very fit and it's not recognized any longer by the immune system, these cells becomes a cell that proliferates and becomes a tumor. So it's like a Darwinian selection. When a cell is so fit that is not recognized anymore by the immune system, this cell, it starts growing, proliferating, and becomes a tumor. There are several treatments that are available. Some are specific for particular mutations that individual patient has. So in other words, when a cell becomes a tumor cell, it has some genetic mutations, and so you can pinpoint these mutations if they are important for the cell to grow, with particular drugs that are specific for that important pathway that leads the cell to a growing cell, so a cell that is growing. And these are treatments that are targeted to that specific mutation. Then you can have, there's some treatments that are affecting all the cells that are proliferating, like chemotherapy, for instance. So chemotherapy is affecting all the cells that are proliferating, including tumor cells. So it's not specific for a tumor cell, but it's acting on proliferating cell. And this is the reason why we lose the hair, for instance, because the hair is made of cells that are continuously proliferating. And so if you are targeting these cells, you are also targeting all the cells that are proliferating. And then there is the immunotherapy. So when the tumor cell is escaping the immune system, so the immune system recognition, you can try to re-educate the immune system to recognize again the tumor cell as a tumor cell, and so to kill it. And this is what is called immunotherapy, and this is what I'm working on. Work on immunotherapy. So I'm trying to educate the immune system to recognize again the tumor cells and to kill them. There are several ways you can do this. One is to reactivate the immune cells. So immune cells have to recognize the tumor cell and to kill it. Okay. The immune cells, including the lymphocytes, T lymphocytes or B lymphocytes, these cells will recognize the tumor cell and will eliminate the tumor cell. The tumor cell, on the other hand, tries to avoid recognition, as I said, and so you have to re-educate the immune cells to recognize and kill the tumor cell. And you can re-educate just by providing some antibodies, which are called immunocheckpoint inhibitors, which can re-educate the immune cell to recognize again the tumor cell. Sometimes it happens that these cells have not been activated at all because the induction of the inactivation of the immune system has started even earlier. So what you can do is you can create therapeutic vaccines. So you can start the immune response to that particular tumor. And so this is a vaccination. And then finally, you can have that tumor cells avoid recognition because they don't express anymore a sort of a flag that tells the body that that cell is a tumor cell. If the flag is not posed on the surface of the cell, then the tumor, the immune system will not even recognize that this cell is a tumor cell. And so what we are trying to do is to re-establish that flag on the tumor cell in order for the tumor cell to be recognized again as a tumor cell. How to re-establish this flag? This flag can be re-established because we noticed, actually many people working in this subject noticed that the patients that were responding better to the immunotherapy had a particular type of microbes in the gut. You know that in the gut we have the commensal flora, which is also called the microbiota. And they were dividing the patients that were responding or not to therapy according to the type of microbiota of microbes that they are harboring in the gut. And starting from this, we said maybe there is something that these microbes are releasing, some metabolites or some mediators that are making the tumor cell more visible and are allowing this flag to be exposed on the cell surface again. And so we have identified a bacterium in particular which does so. So it releases some metabolites which help pointing again this flag on the top of the cell, and so allowing the cell to say, look, I'm a cancer cell, I need to be eliminated. So that's what we are doing. We have identified, now we know that they are bringing up the flag, but we don't know yet exactly how they work. This is important to understand because in case they may not be working then on the patient, at least we know why it didn't work, what could be the steps that were not understood yet and might be important to understand in order to just retail the treatment that we want to make. And the more we know, the better is the efficacy on the patient. What we do is, first of all, we have a library of different microbes that are normally present in our gut. So they are common in our microbiota. We have established a system to have these molecules released. And then we test these molecules directly on tumor cells from different patients and see whether this flag is again exposed on the cell surface. And in this way, we select the bacteria that are most suited for upregulating the flag again, so for exposing this flag. So we do these experiments primarily in vitro. We take the specimens from the patients. We generate tumor cells from the tumor of the patient. And then we put on top of these tumor cells these microbial derived metabolites and see if the flag is exposed or not. And that's how we do. Then we ask ourselves what is within these metabolites, which is the active ingredient responsible for this. And we are at this stage, so we are trying to understand that. Why this is important? This is important because once we identify the exact molecule which is important for re-establishing the flag, this can become a therapeutic strategy to be used in combination with immunotherapy, so to have the two things working at the same time. So the tumor cell putting on the surface the flag, and then the immunotherapy recognizing the cell and eliminating the cell. we should be in this way allowing those patients that are not responding to immunotherapy to become responders to immunotherapy. Just to give the metabolites to the cells, different cells from different patients, because we want to make sure that this is a characteristic which is shared by several tumors, by several patients. And then we will understand what are the pathways, molecular pathways that are activated after we put these metabolites on top. And we do this by several tools, with several tools. One is just to understand what are the genes that are transcribed after the interaction between the metabolites with the tumor cell. So what is the signature which is activated afterwards? In addition, we can also understand if there are some mediators like proteins which are modified after this interaction, and so by proteomic analysis. So the first one was more of a genomic analysis, and now we want to do some proteomic analysis. Altogether, this will allow us to pinpoint exactly what are the molecular pathways which are activated and modified by these metabolites that we have identified. We noticed that these microbial metabolites, they act primarily on certain types of tumors. In particular, we found that it works on melanoma. It works on some hematologic malignancies like lymphomas. But we are quite confident that maybe it's not these metabolites that we have identified, but there may be other metabolites for other types of tumors, so that we can identify metabolites specific for each individual pathology. So for the moment, we have identified for these few pathologies, but I'm quite convinced that in the future, we may be able to identify this for other pathologies. I think that this technology that we have established, or we are establishing, will sooner or later get to the bed of the patient. Now, the problem is that we have to face some regulatory requirements that need to be, of course, addressed, because when you are taking something that is completely new to the patient, it's called a first in man. So it has to go through a series of regulatory activities that have to be done. And this, for us as researchers, is the most difficult part, because this requires some knowledge that we don't have. And so we have to seek help for regulatory agents that will allow us to understand how easy this is to be brought to the patient, because now we have to test this, because it's easier to find this in vitro on a very controlled environment. But then if this really works on the patient, it has to be demonstrated. And so there are three phases in which we do this. The first one is phase one. So phase one means first in man. The first time that you try something on patients, you have to show that this is not toxic, and so the safety of the intervention. Then you have two additional clinical trials that you have to do, which are phase 2 and phase 3. So while phase 1 normally is done on maximum of 20 patients, then phase 2 is done on between 50 and 100 patients. And then you have to demonstrate the efficacy of your treatment in a very high number of patients. And this requires tons of patients. So you have to do this on quite a high number of patients. But then this will be something that other people will be doing. For the moment, we just want to show that the initial activity of these metabolites that we have identified. We arrived now to a point in which we have something that might be working, and we want to keep this to the bed of the patient. So now it's up to now we had starting money for identifying these metabolites. Now we really have to progress, and so we need to bring this to the patient, and to do so we need additional funding. And this additional funding is important because we have to carry out at least the phase 1 to show that these compounds are not toxic. And once we have done that, I'm pretty sure that there will be interest by probably pharmaceutical companies to carry on with this research and then to continue. But we need to carry on our research, because it would be a pity to stop it now. Now, what we need to do is we have to hire an expert for the regulatory part in order to make sure that we are compliant with what the government asked us to do next. And then we need to prepare the metabolites in a way that are suitable for being injected into a patient. And then third, we need to enroll the patients. So we have to discuss with the hospital with whom we want to carry on this phase 1 clinical trial. And for all the bureaucratical work that has been done, in addition, we need the insurance for the patients to be treated. So there are a series of steps that need to be performed in order to continue with this research. We are at a point in which we are very excited, but we need to go ahead.